Okay. So bonjour, hello everybody, welcome you to the second hangout of the MOOC on viruses and human cancers. We welcome you, I'm Hilde, the community manager, and here I introduce the director, Jean-Pierre Vartagnan, to you, and Pascal Pinot, who is a lecturer of that MOOC. And both of you are going to reply to the questions you have been posting in the discussion platform. So actually just a current overview. We have so far 3,122 students from 113 countries, from France, Morocco, Algeria, Tunisia, UK, Belgium, Mexico, and many, many more. So welcome all of you globally, all over the globe. And we currently are in week 7, prevention and vaccination. So if you followed our MOOC on a weekly level, you're already going towards the end. But if you didn't, don't worry, the MOOC is available until 13th of May, so you still have plenty of time to cover it. And now we are going to address your questions. And I read the first question that was asked by Marius. I did not understand very well the course on viral stresses, mutations, and cancers, particularly the role of ApoBex3 gene. Can you please explain it to me?
ApoBex3G or ApoBex3A would be something which is secondary. I mean that if you have a chronic inflammation during 20 years, then you have the upregulation of ApoBex3A or ApoBex3G. In the course, I said that when you have a stress, could be genotoxic stress or viral stress, then you have the release of the mitochondrial DNA, then by different mechanisms will induce type 1 interferon. This type 1 interferon, by mechanism paracrine or autocrine stimulation, will increase the ApoBex3A cytidine deaminase. This protein is an enzyme which is mutagenic. Hence, when you have this upregulation of this enzyme, then the localization could be in the nucleus or in the cytoplasm, and if it's in the nucleus, you will get mutations. And if you have mutation, then you will increase the mutation rate and could be the first step to the tumorigenesis. Hence, you have different mechanisms, but it's the stress that will lead to the upregulation of ApoBex3A will go to the nucleus to make mutations.
Okay, thank you very much. I hope it's clearer now, Marius. The next question I'm going to read to you has been posted by Adalmon about cancer in insects. Thank you very much for your effort in making this topic understandable to simple biologists. From these first weeks, I couldn't catch if cancers are specific to humans or if there are close mechanisms described for other species like plants or other animals. Is there any evidence of cancers for insect species? If there is, are they related to virus infections?
Of course, it's a good question. There is many data in the literature saying that probably the invertebrate can catch the cancer. So it's a complicated question. In terms of mechanistic, in terms of molecular mechanisms, could be the same, meaning that you can induce mutations and then go through proofreading mechanism and then you fix mutations. For example, if you give a mutagen to fruit fly, you put thymidine on the medium, then the fruit fly will eat the thymidine and they will incorporate thymidine. So you will increase the mutation rate. This is described and you can have much more mutations. Now, saying that insect can make cancer, it's complicated questions. There is different parameters. Is it comparable to human? Of course not, because humans have different organs like liver, kidney, stomach, etc., which is absent in insects. So it's complicated to compare cancer in insect and in human. The second point is the numbers of cells. Of course, in the insect, it's small animals with very few cells. So I don't know if you can have enough cells to get, because the cancer starts with one cell. So is it possible to create a cancer in these small animals? The question is open. And the last point I see is the lifetime. So we know that cancer emerges after maybe many years, 10 years, 20 years. And the life time of an insect is very short. So then it's probably complicated to get cancer when the lifetime is very short. So I think that there is different parameters, that there is some point that we can say that the cancer can emerge, of course, in terms of molecular mechanism, but then is it a real cancer? That it's maybe much complicated to answer to this question.
Okay, thank you very much. So actually also Kevin found that question very interesting and has another question. If certain living things do not get cancer, are they being investigated as a possible source of human cancer prevention or treatment? One other thought, seeing how long it takes for many human cancers to develop, do shorter-lived organisms get less or no cancer? I hand over here to Pascal Pinot for the answer this time.
So thanks, Kevin, for the question. It is a complicated question because it is drifting far away from the MOOCs on viruses and cancer. But I will try to answer at least. The first part of the question is there animals who do not develop cancer? In fact, it is considered that all groups of animals develop, if not cancer, at least tumors, as already alluded to by Jean-Pierre. But there is some animals that are more resistant to cancer development. It is naked mole rat, it is some species of bats, it is some bullhead whale, and it is elephant. These different species develop different mechanisms to resist cancer development. It has been published recently that elephants possess in their genome 40 different copies of the p53 genes. Some of them are pseudo genes, but at least they have a lot of p53 genes in their genome. It's a way to escape cancer. It's considered at least to be the case. For the naked mole rat, that is very much studied species. It is a good model for cancer resistance. It is considered that these species develop a specific mechanism of contact inhibition, really sensitive to contact inhibition, and it is depending on the production of hyaluronan. Hyaluronan is a complex sugar composed of glucuronic acid and N-acetylglucosamine that is triggering this contact inhibition. So maybe the use of hyaluronan in therapeutics may at least inhibit or slow down the development of tumors in the future, but it is purely speculative. The other part of your question, Kevin, concerned the lifespan of the animals and their capacity to develop cancer or not. So it has raised a lot of questions a long time ago, and there is experiment of tumor induction that have been conducted more than 20 years ago, where more than 20 species of mammals have been treated by the same carcinogen, and there is mammals like mouse that live 3 years and other mammals that live several decades that have been compared for the very same mutagenic carcinogenic compound, and the researcher concluded that the capacity to develop tumor is not dependent on the normal lifespan duration of the species. It is depending on the dose, the exposure to the mutagenic compound and the mutation that you will develop in your tumor. And in fact, as you know, in mammal at least, the smaller is the mammal, and the shorter is the lifespan. And it appears that bigger mammals are less susceptible than smaller mammals to tumor development, and it has been linked essentially to the metabolic rate of the animals. The small animals have a high metabolic rate. They are producing a lot of heat per gram of body weight. They are eating a lot during the day, and they are producing a lot of radical oxygen species that are considered to be the real mutagenic compound in the tumor development. And in that way, it seems that the smaller mammals are more susceptible to cancer development than the bigger ones.
Thank you very much for the very clear explanation. So now we come to our final question, which has been posted by Mifa. You said it is well known that in patients infected with hepatitis B virus or hepatitis C virus, male gender and an older age influence the risk of cancer. I have read this in some articles, but people couldn't state clearly the reason why. Is there any biological mechanism in men that differentiate them from women? I need more explanation on this point. Thank you very much in advance.
So I will answer this question as well. There is two parts. There is a question of age. So the more you are getting older, the more at risk to develop cancer, any type of cancer, than when you are younger. And it is the case for liver cancer as well. It seems that the risk to develop cancer with age reach a plateau in the 8th decade, after the risk of cancer is not increasing anymore, but you are really old at that time. Regarding now the difference between sex, in fact, at the beginning of the analysis of liver cancer, people believe that the behavior of men is different than the behavior of women. Men are more consumers of alcoholic beverage, and they are more tobacco smoker than women. That's why they will develop more cancer. That was the initial thought of the people. But now it has been modulated, and there is purely biological mechanisms that have been implicated. First, it is the action of hormones. As you know, there is a different balance between men and women. In men, there is a lot of androgens, and in women, a lot of estrogens. And androgens are considered to be more proliferative by essence. In fact, in animal model, male develop much more liver cancer than female animals. And in clinical practice, people who have been treated by androgenic compound develop liver tumors, including hepatocellular carcinoma. But there is so we can say that androgens are proliferative, more pro-proliferative, and that estrogen are anti-proliferative. But there is as well an action of hormones on the immunity. In fact, it seems that the androgen is pro-inflammatory and pro-angiogenic, and it is also the activity of androgen is opposed to the immunity to T cells exactly, whereas the estrogens are more are anti-proliferative, but anti-inflammatory, anti-angiogenic, and they are they are stimulating T cell response, anti-tumoral T cell response as well. The outcome of this different balance of hormones in the different organisms makes that the male are more susceptible to tumor development than women. That's it.
Okay, thank you very much. So we are actually at the end of our Hangout number 2. And please come up with new questions for our next Hangout, which is going to be on 3rd April 2019, also at 3:30 p.m. Central European Time. Have a nice day. Bonne journée. À bientôt. Bye bye.
Bye bye. So.